The overall goal of this procedure is to measure and evaluate the vertical path, velocity, and forces exerted by a freely swimming nematode using coherent monochromatic radiation. This is accomplished by first setting up a coherent monochromatic shadow experiment for freely swimming C.elegans. Then an individual adult C.elegans is placed into the shadow experiment viewing chamber, which is a cuvette. The shadow of the worm as it descends through the laser beam is recorded as a movie, and the movie is used to analyze the motion of the animal. Hence, the path of a freely swimming nematode and the associated forces generated are determined.
The main advantage of this technique over existing methods such as traditional microscopy in a horizontal plane is that the motion of freely swimming microorganisms can be investigated using a wide range of monochromatic light at a low cost.
This method can answer key questions about forces generated by or responded to by nematodes in a liquid environment.
We first had the idea for this method when we first started exploring the idea of vertical movement for microscopic species.
Visual demonstration of this method is critical, as the procedural steps are difficult to learn because it takes some practice to get the laser alignment correct. Furthermore, some time in the biolab is necessary to learn how to prepare the worms.
Assemble the experimental setup to create shadow images. Use 2 or more mirrors to steer the helium-neon laser into the Galilean beam expander, where it should increase to 12 mm. Have 1 or 2 pinholes in the beam path to ensure its alignment. From the expander, direct the beam to a mounted quartz cuvette.